Hey there, welcome to part two of this three-part lecture series on metabolism, microbial metabolism, but metabolism in general, the basics of the metabolic processes and the pathways involved within cells. So we're going to pick up here with beginning to look at the reactions, how electrons are involved, and see the progression of these metabolic reactions that occur within cells. But before we get into looking at some of the pathways that are involved, we need to cover oxidation reduction. The terms oxidation and reduction are applied to electrons and whether or not they're being added to or taken away from something. Oxidation is the removal of electrons. So when a molecule or an atom is oxidized, electrons have been removed. Whenever an atom or a molecule or a compound is reduced, it's gained electrons. And that's what these processes are based upon. Compounds picking up electrons, compounds giving off electrons. Picking up, giving off, picking up, giving off. So they're often paired together where these oxidation reduction reactions are together like Oreos and milk. And so we call them redox reactions. The re for reduction and the ox for oxidation. So a redox reaction is an oxidation paired with a reduction. And a way to help you remember that, a little mnemonic device is Leo the lion goes grr, where we have Leo, lose electrons oxidation, and the grr part, gain electrons reduction. Now, I guess Leo the lion, I mean, he's a lion, he's not going to go grr, but I guess it helps. I don't know, but we can get past that if you're not too particular about this, about the sounds that lions make, and you can get past that. Then Leo the lion goes grr is a mnemonic that will work just fine for you. So lose electrons oxidation, gain electrons reduction. And so what this picture down at the bottom is showing that you've got A has an electron, well, it gives its electron to B, A becomes oxidized, B becomes reduced. It's as simple as that, just giving up and receiving electrons, redox reactions. In biological systems, the electrons are often associated with hydrogen atoms, so hydrogen becomes the electron donor often, or the electron acceptor. So biological oxidations are often what are called dehydrogenations. And so what this shows here is you have an organic molecule, that's that yellow blob there with those two, they look like googly eyes sticking on the top of it. Those are the hydrogen atoms, and hydrogen atoms, as you know, are composed of just one proton and one electron, no neutrons. Well, what they often do is they will be transferred to a coenzyme. Coenzyme, one example, and another one we're going to look at is called FAD, but this one's called NAD+. There's one involved in photosynthesis called NADH+. And so what happens here is that these hydrogens, along with their electrons, get transferred. So that organic molecule that has lost electrons has been oxidized, and the NAD+, which has gained a proton and two electrons, that has become reduced. It's a reduced electron carrier. Remember, lose electrons oxidized, gain electrons reduced. And then one hydrogen proton, which is designated as an H+, is left for other purposes, which there's a lot that could be said about that, but we're not going to. We'll just keep it very simple and just say that H+, that proton, is used for other purposes within the cell. So we're going to be talking about this, how these organic molecules like a carbohydrate gives up its electrons and becomes oxidized, and how the coenzymes, these electron carriers, become reduced. And that's the basis of these biological reactions that occur within the cells. So I'm taking it in slow, small steps, baby steps through this. So we learned what oxidation reduction reactions are. Here's a small example of how this happens. You have an organic molecule giving up its electrons to an electron carrier, because you don't want those electrons are going to be used for something else later in another process, and it's a variety of processes. So NAD+ will hang on to them, to those electrons to be used elsewhere. So we'll talk about how is ATP generated. There are different ways that it can be made. Remember, ATP is the cash money energy molecule for the cell. You start with ADP, which is adenosine diphosphate, and you can see you got two phosphates there. Add some energy in a phosphate molecule, an independent one, and you end up with adenosine triphosphate, or ATP. So that energy gets locked within that bond between the second and the third phosphates. And that energy can be utilized by the cell when that bond breaks through the use of enzymes, breaking that bond, releasing the energy, and then using that energy to power cellular processes like active transport or phagocytosis or making new organelles or whatever. On and on it goes. So we take in, say, for example, our foods, we consume them, the foods get broken down into their smaller constituent molecules and so forth, and then the energy that's contained within the bonds of our food gets transferred from there to ADP. ADP becomes ATP, and now the cell can actually use this form of energy. So in the form of the molecular portions of our food, say when we eat like a bowl of Lucky Charms and we get all this carbohydrates and sugars, well, they can be broken down into glucose, but the cell cannot use glucose. Glucose has way too much energy. One molecule of glucose has way too much energy to be utilized by a cell, because remember, a cell is very small and tiny, and so the processes that happen within it are also very small and tiny, and so they need an energy molecule that will give them enough, but not too much, that it's an overabundance of it, and that can create problems within the cell, like raising the temperature, too much energy being released at once, too much exergonic reactions occurring, too much heat gets released, temperature goes up, enzymes denature, cell dies. So this is an energy molecule that has a very small amount of energy, and it's very versatile and will give up that energy rather easily. And we can use billions of them at a time in order to carry out some cellular process, or if a muscle fiber needs to contract, for example, billions of ATP molecules can be utilized to make a muscle fiber contract. So there's what's called substrate level phosphorylation. The process phosphorylation, it sounds so geeky, but what it is it's just taking a phosphate and adding it to something. So at the substrate level, it's just a transfer of a high energy phosphate, which is PO4 negative, to ADP. So you've got you start with the ADP, you've got the phosphate with the energy attached to it, add that to the ADP, and you simply get ATP. So that's substrate level phosphorylation. There is what's called photophosphorylation. So this is another way to generate ATP, but in this case we're using photo, which means light. So light causes chlorophyll to give up electrons. So the chlorophyll molecule becomes energized by light energy. Those excited electrons up their energy level, and then they are transferred through a series of electron carriers that pass along the electrons. These are all redox reactions. They gain electrons, they lose electrons, they gain electrons, and that's what that little stairway thing is trying to show you there. You're going down the steps. It's not a stairway to heaven, it's a stairway going down, I guess, for you Led Zeppelin fans. And then what happens is the energy then is released in small amounts, and that energy then is utilized for the production of ATP. And so this is why it's called photophosphorylation, because light is necessary in order to make these photosynthetic processes actually occur, which we'll talk about at the end of the chapter briefly. So let's look at carbohydrate catabolism. So the breaking down of carbs. And there are other, you can look at the metabolism of lipids and proteins and nucleic acids, but those are usually ancillary. They're usually put at the end of an introductory chapter on this topic. And if this was a biochemistry course, then we would look at them in more detail. But typically, to introduce the concept of metabolism, we look at just specifically carbs. So what is it? It's the breakdown of carbs to release energy from them, and you can take two very different pathways. Aerobic, meaning oxygen is involved, and anaerobic, meaning there is no oxygen involved in the process. The three pathways of aerobic are glycolysis, Krebs cycle, and the electron transport chain. The two pathways of anaerobic are glycolysis and fermentation. of which we'll look at lactic acid fermentation and alcohol fermentation. There are other types of fermentation, but those two are by far the most common. So let's begin with the aerobic side. We're going to start with that. They both begin with glycolysis, so let's look at this one. So split the word in half, glycolysis. So this means the splitting, the lysis part of glucose. The GLY refers to sugar, so it's the splitting of sugar, in this case the splitting of glucose. So glucose will become oxidized. In other words, it's going to lose electrons and it's going to give up its energetic electrons and pass those electrons on to electron carriers. And it'll get split in half into a compound called pyruvic acid, or just simply pyruvate. It produces a net total of 2 ATPs, so small energy molecule production, and 2 NADHs, which are the electron carriers. Those are the ones that will hang on to the electrons, but will not leach the energy from those electrons that came from glucose. So all these processes begin with glucose and the splitting of glucose and extracting the energy from glucose, basically extracting the electrons from the glucose. So in this slide, this is what's called the preparatory stage. And in the preparatory stage, the glucose gets split at the very end. It's split into 2 3-carbon compounds. Now, my college students, you're not going to need to know each and every one of the products, the glucose 6-phosphate, the fructose 6-phosphate, and so forth. All I want you to see is that in this case, in the preparatory stage, number 1, glucose is split from a chain from a 6-carbon compound to 2 3-carbon compounds, and ATP is utilized. So ATP is used twice. So one ATP molecule between steps 1 and 2, and then another ATP molecule between right there at step 3. So what happens is the phosphate is attached. It gets attached to the carbon chain there, as you can see in this graphic, and then it becomes ADP. So ATP gives up some energy. That energy then is locked in that bond of the phosphate, and it's added to the glucose molecule. So this is what's called the preparatory stage. And you'll notice at the bottom, those two compounds, there's a double arrow there. So when the glyceraldehyde 3-phosphate moves on, the dihydroxyacetone phosphate will get shoved over, and then it will continue on in the chain. But it only handles, after this point, just half a glucose molecule at a time. And again, we have a series of steps, and then we end up finally at the end with pyruvic acid. So 2 glyceraldehyde 3-phosphates are oxidized into 2 pyruvic acids. Eventually, that's what happens to them. So the pyruvic acid is the final product of glycolysis. 4 ATPs are produced grand total. As you can see here, 2 ADPs at step 7 become 2 ATPs, and at step 10 the same thing happens. But only 2 can be used by the cell because in the preparatory stage 2 ATPs were utilized. So we have to take 4 minus 2, and so we have 2 usable ATPs that are left, and 2 NADHs are produced. Those are the electron carriers. So essentially the glucose, or the specific molecule in the chain, becomes oxidized. It loses its electrons and gives them to NAD+. NAD+ becomes reduced into NADH because it has gained electrons. And now finally at the end we have pyruvic acid. Now pyruvic acid then can go in either direction depending on if it's going to be aerobic or anaerobic. And my college students, you can find this PowerPoint, and then you'll be able to click on that hyperlink which animates glycolysis. It shows it in an overview and explains it pretty well too. So let's look at cellular respiration. We're going to be talking about the aerobic side here. Oxidation of molecules liberates electrons for an electron transport chain, and ATP is generated by what's called oxidative phosphorylation. So this all has to do with the loss of electrons and the gaining of electrons. So at the end of the process, through all these redox reactions, a whole bunch of ATP is produced as a result of it. So we'll go with the Krebs cycle. And remember, this is the aerobic side of things. If this were the anaerobic side, we would be looking at fermentation right now, but we're going the aerobic pathways, where oxygen becomes the final electron acceptor. And that's why it's called aerobic. And you'll notice in the Krebs cycle, you're not seeing any oxygen being utilized here. So that comes in the next pathway called the electron transport chain. So what happens is we pick up right where we left off with pyruvic acid. Now, in a bacterial cell, this is all happening in the cytoplasm. If this were in a eukaryotic cell, this would be happening, the Krebs cycle would be occurring within the mitochondrion. So there needs to be a transfer of the pyruvic acid from the cytoplasm into the mitochondrion. So in all cells, and I should have stated it this way, glycolysis occurs in the cytoplasm, prokaryotic and eukaryotic. That's where glycolysis is taking place, out in the cytoplasm. But with prokaryotic cells, they don't have mitochondria, so Krebs is taking place in the cytoplasm. In eukaryotic cells, they have mitochondria, so Krebs takes place in within the mitochondrion. So pyruvic acid is oxidized, it loses electrons and becomes the compound called acetyl-CoA, and carbon dioxide is given off. With this, you'll notice also on the left side of the diagram, NAD plus becomes reduced and becomes NADH. So pyruvic acid is oxidized, it gives up electrons. Well, what does it give up its electrons to? It gives up its electrons to NAD plus. So NAD plus picks up the electrons and becomes NADH. Then acetyl-CoA is further oxidized, so it loses a carbon molecule through the molecule a carbon atom through the molecule of carbon dioxide. And carbon dioxide then is a waste product. That carbon dioxide leaves the cell. In a bacterial cell, it'll just go into the extracellular environment. Within us, it'll go into, it'll leave the cell and eventually be picked up by our blood cells and then to our lungs, and then that's what we exhale. So that becomes a waste product. And as you know, the more you exercise, the more you inhale, the more you exhale as well. You produce more carbon dioxide. Well, why do we do that? Because when we're exercising, we are kicking up all of these processes many times more, and so therefore more carbon dioxide keeps getting belched out of the Krebs cycle, and we have to get rid of it. And so we start breathing more heavily. We're craving more oxygen, so we need to take in more air, greater amount of inhalation, but at the same time we're creating more carbon dioxide, and we don't want that to build up, which that can be deadly, and so we exhale. And as I said, NAD plus is reduced. It picks up electrons, energized electrons, and FAD2, it's another electron carrier, which you'll see in a bit here in the next steps of the Krebs cycle. It gets reduced to FADH2. And notice they're both picking up hydrogen atoms as well. There's a generation of 2 ATPs per molecule of glucose in the Krebs cycle. Now you'll notice in this prep stage here, there's no ATP seen. That's in this part. This is actually the cycle part. And there's a picture of the man himself, Hans Krebs, who was the one who kind of described and elucidated this process here and laid it out for us. He won the Nobel Prize for it, and deservedly. And nobody else was even really able to figure all this out except for him. So working in his lab here in the United States, in Great Britain, he figured out the Krebs cycle. So you can see we have an 8-step process here, and it just changes like you saw in glycolysis. One compound becomes the next compound, becomes the next compound. But what I want you to see in this cycle is, notice that we start out with citric acid, and that's why it's also called the citric acid cycle, because citric acid is the molecule that kind of begins the cycle. However, it's a cycle. It can start anywhere, but we kind of are coming down right there. So we're taking the acetyl-CoA and we're joining it to the citric acid, we're joining it to the oxaloacetic acid. If you look on the upper left, that's a 4-carbon compound. It comes around, joins the 2-carbon compound from the prep stage, and now we have what's called citric acid. Then things change a bit. NAD plus becomes reduced to NADH. Carbon dioxide is given off as a waste product. So we had a 6-carbon compound. Now we're down to a 5-carbon compound called alpha-ketoglutaric acid. Then the same thing happens again. Another NADH is shot off, and another carbon dioxide molecule is a waste product. Now we're down to a 4-carbon compound called succinyl-CoA. Then ADP becomes ATP, it's phosphorylated, and then we have succinic acid moving upwards now, vertically heading up. FAD becomes reduced to FADH2. Then we have fumaric acid. Then that gets transformed into malic acid. NAD plus becomes reduced to NADH. Then we have oxaloacetic acid. And then it keeps going around again. Now, you'll notice there's only one ATP, but the previous slide I said there were 2 ATPs. That's per molecule of glucose. Remember, this started with basically, it was a half a molecule of glucose. Pyruvic acid is half a glucose. So one pyruvic acid goes through, you get 1 ATP, then the other half of the glucose, the second pyruvic acid goes through, and you get another ATP. So what you're looking at here basically is half a glucose. If you want to get a full molecule of glucose, you just take all these and double them. So rather than 4 NADHs being produced total, you get 8 NADHs produced altogether. Instead of 1 FADH2, you get 2 FADH2s, and instead of 1 ATP, you get 2 ATPs. And I might as well mention the carbon dioxide because we're familiar with that. Instead of 3 carbon dioxide molecules, there are 6 carbon dioxide molecules produced per molecule of glucose altogether. Those are the products of the Krebs cycle, and CO2 is the waste product. It's not utilized anywhere else in the process, but all the other ones will be moving on to the electron transport chain. So the electron transport chain is where the boatload of ATP is produced. The NADH and the FADH2 from Krebs become oxidized. They're now going to lose their energized electrons. Where did those electrons ultimately come from? From the glucose. From the carbs that we ate, our digestive system breaks down the complex carbs into the simple sugar glucose. The glucose then becomes the beginning of glycolysis, and the Krebs cycle then follows, and then the ETC follows after that. So all these electrons, they initially came from glucose molecules. This is done by removing the electrons and passing them through a series of cytochrome. There are a bunch of enzymes, carriers like a hot potato, so they pass them on. So one of them gains an electron, so it becomes reduced, and then it gives up the electron, it becomes oxidized, and then the next cytochrome becomes reduced, and then it gives off the electrons and becomes oxidized. So it's just a reduction, oxidation from one enzyme to the next. The final electron acceptor is oxygen, and that's why we call this process, we describe it as being aerobic, because oxygen is involved, which then joins with H+ ions to form water, which is a waste product. And so that water then can be utilized by the cell, or it just enters the bloodstream, and then it eventually makes its way to the kidneys, gets filtered out, put into the bladder, and then we excrete it from the body. Or if we're working out heavily and it's hot, that water will come out of the bloodstream and into sweat glands to cool us down. So this water here that we're excreting or sweating out, that's coming from the electron transport chain as a waste product. In prokaryotes, there is a total of 34 ATPs that are generated. In eukaryotes, there are a total of 32 ATPs that are generated. This has to do with the mechanics within the cell. Prokaryotes don't have to deal with mitochondria and transporting things through membranes, and so they're able to generate a couple more ATPs compared to eukaryotes. Now, these numbers are not tried and true. They vary. Not every cell is going to produce exactly 34 ATPs in a bacterial cell or 32 in a eukaryotic cell. It varies from one cell to another, even from organism to organism, but roughly speaking, that's about the average number of ATPs that are produced. The energy released from the electrons is used to produce ATP by a process called chemoosmosis, moving electrons through a membrane. So what it kind of looks like, this is what I was describing earlier, where you have a reduction and an oxidation. They keep just moving the electrons bit by bit from one carrier to the next, from one carrier to the next. They're not all released at once. That would be devastating to a cell. It would be too exergonic. It would destroy things within the cell. So the electrons and their energy is given off bit by bit, and the flow of electrons moves down through these electron carriers. Their energy is utilized in order to do this. So this would be from a eukaryotic cell. because if you look at the left of the picture, the inner mitochondrial membrane, what's happening is the electrons are given off and then they're passed from one carrier to the next. And as the electrons move down through the chain of carriers, these electron carriers, the energy from the electrons is used to actively transport H+ ions. So those H+ ions are floating around inside the mitochondrial matrix. If this were a bacterial cell, this would just be in the cytoplasm. Where did the H+ ions come from? They come from NADH. NADH splits. It loses its hydrogen. That hydrogen ion then becomes liberated and floating around. Those hydrogen ions get transported through the membrane, and one side of the membrane, in this picture, the upper side of the membrane, becomes full of positive charged protons. And this creates an imbalance of charge. And when that happens, we have a force that's generated called the proton motive force. And as a result of that, something can happen. There are a number of things that can happen as a result of an imbalance of charge between a membrane. And the proton motive force can be utilized for a number of biological processes. In this case, it's going to be used to make ATP. So in the mitochondrial membrane, the inner membrane, there's an enzyme called ATP synthase. It's at the right of this diagram, kind of in blue. And what happens is those hydrogen ions, those protons, cannot get through the phospholipid bilayer of the mitochondrial membrane until they reach ATP synthase. And when they reach ATP synthase, zoom, they can go down through that enzyme, and what happens is the enzyme spins, it grabs an ADP and a phosphate, joins them together and makes ATP. So this is an enzyme. Look at its name, ATP synthase. It ends in ASE, which means it's an enzyme. So this is how the process happens. This is how all the ATP get generated. Now, if this picture showed a bacterial cell, erase inner mitochondrial membrane and just call that the cell membrane. So in bacteria, they have their cell membrane, but remember, there's something on the outside of that. There's a little space, and then remember, they have the cell wall. So that space that's called the periplasmic space. That's where this would be happening in the cell membrane, and those hydrogen ions, those protons, would not just float away into Never Never Land. They would be held in that space by the cell wall of the bacterial cell. And within the cell membrane of the bacterial cell, there is also ATP synthase. So this all looks pretty much identical in the bacterial cell, other than the fact it's not in the mitochondria because they don't have mitochondria. So this is the ETC, active transport of H+ ions across what are called proton pumps, the return of those H+ ions through ATP synthase. So this is like a big circle for the protons. They get pumped through the membrane, they fill up on one side, then they shoot back through the membrane through ATP synthase. They provide the force that's necessary for ATP synthase to generate or synthesize ATP. I explained the location in eukaryote. This is happening, as you see in this picture, in the inner mitochondrial membrane and the matrix of the mitochondrion, which is like the inner guts of the mitochondrion. And prokaryotes, that would be the cell membrane. And instead of the mitochondrial matrix, you would have the cytoplasm instead. I have greatly simplified these processes. It's some pretty heavy stuff here. There's a lot more details that I just glossed right over. But again, this is introductory microbiology. This is not some biochemistry course for biochem majors, so we don't get into the details of those. But I wanted you to have an overview of what happens within these processes and to give you a better idea of them. So I'll end there for part 2. Part 3 will be coming up, so stay tuned for that one, and thank you for watching.